Hey everyone, welcome to your proteins debrief. I'm also going to go into helpful information about how the structure of proteins is going to affect its function. And these notes we are going to write in this top half of your mega sheet. So this is just the crease. It's not an actual line. I'm going to probably start here close to this information and then just move all this way. So on this side, you have the background information about proteins. They're made of carbon, hydrogen, oxygen, nitrogen. Their monomers are amino acids. The bonds holding the amino acids together are called peptide bonds. They have many functions, including catalysts, structure, defense, movement, transport. Proteins are a large class of molecules that have a diverse function within living cells. So again, I'm going to write this information across this top half of the mega sheet. So in this video, our objectives are to describe the structure of an amino acid, to explain how dehydration synthesis makes proteins, and to describe the levels of protein folding. We're going to talk about what the levels of protein folding are, and we're also going to talk about the bonding type for each level of protein folding. We're going to start with describing the structure of an amino acid. So again, this is on the top portion of your mega sheet, right next to where you wrote down the basic information about proteins. So when we're describing the structure of an amino acid, we always begin in the center of the molecule. And the center of the molecule is going to be a carbon. Now I recommend you don't write this down until I'm done drawing the complete molecule. At that point, I will go ahead and say to stop the video so that you can pause the YouTube video. That way you can focus on learning the material and then copy it down once I have completed the drawing. So our amino acid starts with a central carbon, and the carbon has 4 valence electrons. And so what we're going to do is we are going to draw 4 bonds around this central carbon. Now, for most amino acids, 99% of the amino acids, they are going to contain a hydrogen group attached to one of these bonds. And so I just drew it here at the top, but sometimes you'll see it here at the bottom as well. Next, around the central carbon, we are going to draw a carboxyl group. So a carboxyl group is carbon, oxygen, and then a hydroxyl group, OH. So this COOH is the carboxyl group, and this is what its structure looks like. So you have a carbon double bonded to an oxygen, and then the hydroxyl group coming off to the side here. Next is the amine group, and the amine group is NH2. So nitrogen and then 2 hydrogens, that is the amine group. The last group of the amino acid is given a generic letter representation called R. And the R group is different for each of the 20 amino acids. Some of these R groups can be hydrophobic. Some of them can be hydrophilic. Now, sometimes these R groups can also be described, instead of as hydrophobic, they can be described as non-polar. And instead of hydrophilic, they would be called polar. The difference between these two is that these terms are referring to its ability to interact with water, whereas these terms are referring to whether or not they have a positive or negative charge to the molecule. But polar molecules are hydrophilic because they can interact with water, and non-polar molecules are hydrophobic because they don't interact with water. Now go ahead and pause the video and draw this in your mega sheet. Now that we've described the structure of the amino acid, we're now going to use that structure to explain how dehydration synthesis makes proteins from the monomers of those amino acids. So again, wait until I finish drawing before you copy this in your notebook. So I've drawn two different amino acids. So here's the central carbon. Here's that R group that makes this amino acid different from this one. That is the carboxyl group, and that is the amine group. This is the second amino acid. This one has a different R group attached to it, but it still has that central carbon, the carboxyl, and the amine group. So when dehydration synthesis occurs, remember that we are going to make water, H2O. And you'll notice that on this amine group, there's no oxygen that's going to be available to donate to this dehydration synthesis process. So the carboxyl group on this amino acid is the one that's going to give up this OH group. And this amine group on this side is going to only need to give up one of the H's. So this is where the water is going to come from. We've got the OH group coming from this carboxyl group, and we've got the hydrogen coming from this amine group on this other amino acid, and it's going to be removed to produce water. Then what's going to be left behind is a bond that can form between this carbon valence electron and the valence electron of this nitrogen. Remember that those valence electrons are now free when we remove the water. So when we redraw this dipeptide, called a dipeptide because we've got two amino acids, we get the formation of a dipeptide bond shown right here between this carboxyl group and this amine group. So this dipeptide, di means two, is showing you the bond between the carboxyl group of one amino acid here and another amino acid over here on this side. So the R groups you can see are the same from up here. This is that central carbon. And the two amino acids are held together by this peptide bond that occurs between the carbon and the nitrogen. Go ahead and stop the video now and record this in your notes. The last topic we're going to talk about is protein folding. And the easiest way to do this is by doing a comparison chart for the 4 different levels of protein folding. Once again, I recommend you wait until I tell you to stop the video to record this in your notes. All right, so we're going to compare the levels of protein folding, beginning with the primary level. Many times you'll see this written as 1, and then with the circle indicating that this is the prime level or the first level of protein folding. We can also think of this as the protein's structure. So the primary level of protein folding is the order in which the amino acids arrange themselves based on the DNA. So you can think of proteins and the amino acids like beads on a string, and the specific order in which the beads come in, so they can be different shapes, different sizes, different colors, but the specific order that they have to line up is what's known as the primary level of protein structure. And so in this video, you're going to see me refer to AA very often, which stands for amino acid. Now, the bonds that hold the primary level of protein structure together, the specific order in which the beads are, is what's known as that peptide bond. You may remember from 9th grade the process of transcription and translation. Well, translation is this primary level of protein folding that is held together by peptide bonds. So this is what you did in 9th grade when you did translation. But the protein continues to have additional structure, including the next level, which is called secondary level structure. Now, for secondary level of protein structure, the protein or the beads on the chain start to develop a secondary structure level based on the hydrogen bonds that form between the amino acids. Now, in this case, that structure will either be an alpha helix or it will be a beta pleat. And again, secondary structure is held together by hydrogen bonds. Now, tertiary structure is where things get really, really complicated, because tertiary structure is now going to incorporate all the amino acids that are hydrophobic or nonpolar and bring them together so that they avoid the aqueous environment that the cell is in. And so it takes on a very complex three-dimensional structure. And so we focus, when we talk about those tertiary levels of protein folding, what our focus is on those hydrophobic interactions. And it's not just going to be hydrophobic interactions that are holding this structure together. It's going to also be hydrogen bonds. You can get ionic interactions, which is between a positive and negative. You can also form something that are called disulfide bridges. Di means two, helping out in this process. Now, the last level of protein folding is what's called the quaternary level of protein folding. And in quaternary level of protein folding, this is where you get multiple chains coming together to form one functional protein. So you have multiple chains of peptides, what we call peptide strings, coming together to form a globular or even more complex protein structure. So a very common example would be hemoglobin. Hemoglobin is made of 4 peptide chains that come together to form that one protein, like this. And so you can see you've got 1, 2, 3, 4 chains that are coming together to form one hemoglobin protein. And the same types of bonds that are in tertiary are going to be in quaternary. So we'll have hydrophobic, we'll have hydrogen bonds again, we'll have ionic interactions, and we'll have disulfide bridges. This concludes the information on proteins. Remember, in this video, we wanted to describe the structure of an amino acid, explain how dehydration synthesis makes proteins from amino acids, and then describe how that protein folding or protein structure in terms of what it is and the bonds that were involved. I hope this video was helpful. I'll go ahead and end the video without the objectives on the screen so that you can copy this down in your notes. Thanks for watching. Bye.